Hello everyone, I'm Mohammed Alser, and today I'll present about accelerating genome analysis, which is our own journey to accelerate this important step. And I'm a senior researcher and lecturer of bioinformatics and computer architecture at ETH Zurich. So, to analyze the genome, first we need to read the content of such genome. Unfortunately, we don't have any machine that gives you the complete sequence of a genome as output. What do we have is a machine called sequencing machine that provides small randomized fragments of the original DNA sequence. And these fragments, we call them reads. And to get useful information out of these fragments, we need to analyze or link these reads or put these pieces together. And that is what we call a computational step. We call it read mapping. And when we analyze the performance of sequencing and genome analysis, we find that these sequencing machines are very capable and they can generate, they can sequence a large number of genomes, like 48 genomes at 30x coverage in about only 2 days. And when we look at the genome analysis, we find that this is very less than the throughput of the sequencing, which can analyze only single human genome in about 32 CPU hours. That is using 48 core processor. Of course, these numbers might vary from machine to machine, using different data, different setup, and so on. But this trend, it's still the same, especially with the Moore's law is going to saturate very soon. And for that, we really need for speed. And there are many applications that are life-threatening, and we really need very fast whole genome sequencing to address these problems. For example, if you are looking for costly whole genome sequencing, you could get it in 2 days. That is the entire duration until the interpretation, or less costly, that is 5 days. And if you do the fast one, then you could avoid morbidity for critically ill infants or reduce both the cost and the length of the hospital stay, as shown in this research. You can think about many other applications that really require very fast genome analysis and then changing the management during the hospital stay, such as blood sepsis and many other diseases. And it's not only about the time that genome analysis takes, but also the scalability. How capable are you to do it for thousands of patients in the same time, for example? So that is the case in the UK, where they start doing whole genome sequencing for all seriously ill children admitted to hospital. And the same thing applies to the very large studies, such as population scale microbiome profiling. This is a recent study done this year where they try to do metagenomic analysis for 60 cities. And there are many more applications that require not only to do sequencing, because sequencing is done once, but also you store the data somewhere in a publicly available database where large studies can download this data and then do analysis again and again. And for these studies, you really need to do fast and scalable genome analysis. All right, now what makes read mapping a bottleneck? First, we have Tsunami of sequencing data. Remember that read mapping is the very first step after base calling, where the entire data coming from the sequencing machine will be handled or processed first by read mapping step alone. And unfortunately, we are still using general purpose machine for the analysis, where in the sequencing, we have sophisticated machine that do just only sequencing, which is very fast compared to the general purpose computing. And as we are using traditional architecture for computing, the sequencing machine will transfer the data to the storage, and then the storage to the main memory. From the main memory, move the data to the several level of caches, all the way to the processor or the GPU or FPGA. And with that, we have a huge amount of data movement between these several components. And remember from computer architecture class that single memory requests consume at least 160x more energy compared to performing an addition operation in the processor. So requesting data from the memory is always expensive, and data movement always dominates performance and is a major bottleneck, major energy bottleneck in such systems. And when we do the analysis for the execution time for Minimap2, for example, we find that more than 60% is spent in sequence alignment. That is not to include, for example, filtering and all the helping steps that help the sequence alignment to perform better. So sequence alignment is done using dynamic programming, which is very expensive to perform and very expensive also to accelerate due to the data dependencies in this step. And also in read mapping, we have a large search space for mapping location. We are searching the entire reference genome to find a few locations that may fit, say, a given read. All right, for that, we need an intelligent algorithms and intelligent architecture that handle data very well. And this is what we are trying to survey in our paper, where we find a large number of algorithms that try to accelerate every single step in the read mapping or one or more of these steps. And we find that there are three main steps. The first one is indexing and seeding, and the second one is pre-alignment filtering, and the third one is sequence alignment. We can find exact algorithm. We can find also heuristic algorithm that try to compromise the accuracy to gain some speed up. In our research group in Safari at ETH Zurich, we tried to tackle this problem by proposing a new algorithm plus a new architecture. And we did that at different level of different component of the compute system. For example, we started with specialized pre-alignment filtering accelerators using GPU and FPGA. And we have several works, such as Gatekeeper, Magnet, Shuji, Gatekeeper GPU, and SneakySnake. And then we move further and try to do the processing near memory without the need to move the data further away from the main memory, such as gram filter, SneakySnake, and GenASM. And then we try also to accelerate sequence alignment using GenASM. That is done near the memory, where we reduce the data movement trip from the main memory to the processor. So the first one was try to do specialized hardware for pre-alignment filtering. And we observed that correct alignment is a sequence of non-overlapping long matches. And the key idea is that to approximate edit distance calculation based on finding these non-overlapping long matches. And that is very similar to a single net routing problem in VLSI chip. This is the VLSI chip, and this is after we dissect such a chip and we try to see the component inside it. You can see the legs of the chip over there to the left and the right side, and the trip from the left side to the right side through this component is what we call a single net routing problem. And we try to solve it by moving in straight lines, and whenever we face an obstacle, then we detour to a different straight line, and then we repeat this step. And the number of obstacles we face through this path is the lower bound on the edit, the number of edits we have for these sequence pairs. And that is a very fast way to filter out a large number of genomic sequences. So the key results we got from SneakySnake is very significant. And we can see that we got order to 2 orders of magnitude speed up compared to state-of-the-art sequence aligner, such as Idolib, Parasale, and KSW2 for both short and long reads. And now we try to do this SneakySnake algorithm or processing near memory. And this is a recent work we published last few months. And we noticed that read mapping is heavily bottlenecked by data movement from the main memory. So we said, why not to do the processing near the memory or very close to the main memory where the data resides. And we carefully redesigned the accelerator logic of SneakySnake to exploit such near memory computation. And we use for that this FPGA board where we have this FPGA chip, and it's integrated on the same package substrate with the high bandwidth memories. And the improvement we got based on our design, we see very significant performance and energy efficiency improvement by at least 27 and 133x respectively, compared to 16 core or 64 hardware threads IBM Power9 CPU. And for the near memory sequence alignment improvement, Our goal was to accelerate approximate string matching problem by designing a fast and flexible framework which can accelerate multiple steps of genome sequence analysis. And we have three key ideas. The first one is to exploit the high memory bandwidth and the logic layer of the 3D stacked memory to perform high parallel approximate string matching. And we modify and extend a BiTab algorithm that is a very old algorithm, was proposed in 1992 to do approximate string matching with fast and simple bitwise operation, which makes it very suitable for hardware accelerator. However, it was limited, and we extended its capabilities such as supporting long read, supporting traceback, and make it parallelizable, such as we can divide the problem into subproblems. And then we co-design our modified algorithm with the new hardware architecture, try to build low-power, area-efficient hardware accelerators. And we use the same architecture for three different use cases: read alignment, pre-alignment filtering, edit distance calculation. And the experimental results were really significant compared to Minimap2, BWA-MAM, Darwin, and GenAX, which are hardware-accelerated read mapping. And also in terms of pre-alignment filtering, edit distance calculation, we get an order to 2 orders of magnitude speed up, plus energy efficiency, which is very important in integrating the hardware accelerators with sequencing machine, for example. We don't want to overheat the sequencing machine, for example, or being power hungry. So the adoption of these hardware accelerators in real genome analysis, it's not new. For example, an Illumina company started in 2016 to adopt these Dragon FPGA boards to do the analysis inside the sequencing machine, for example, or using NVIDIA GPU boards to accelerate genome analysis. And the results, you can see, it's significant. However, they're still suffering from data movement as they are not near memory or near storage processors. And they still move data between different components based on the steps or the pipeline dedicated for that. They try to tackle this using some hardware component dedicated for data compression, decompression, but still the effort there is limited. And there are many more challenges for adoption of these hardware accelerators. The first one is to accelerate the entire read mapping process rather than its individual step based on Amdahl law. So accelerating single step and then try to integrate that with software-based read mapping would not provide great benefit as if we accelerate the entire process. And we still need to reduce the high amount of data movement. For example, compression is good, but if we can develop new algorithm that can work directly on compressed data, that will enrich such hardware accelerators. And it will be always the best solution is to filter out unlikely reused data at the very first component of the compute system, which is the storage, for example, or inside the sequencing machine itself. If you know that this read is not going to map anywhere, then it will be much better or more efficient to filter out such read inside the storage rather than moving it to the main memory all the way to the processor. And such hardware accelerators really need to provide a wide range of values for each of the parameters used in read mapping rather than limiting these values. And we need to adapt existing genomic data formats for hardware accelerators or develop more efficient file formats that can really reduce the data movement. For example, in FastQ file, each character is represented using single byte or sometimes 2 bytes. So it doesn't make sense to move all these data to the FPGA to do the processing. We normally use 2-bit encoded data or 4-bit encoded data. And that is done in Minimap2 internally, which also wastes some of the execution time. So it will be efficient to really adapt new file formats that are, let's say, hardware friendly. Also, we don't want to compromise the accuracy at all. So they use some heuristics to gain speed up. That's not going to go anywhere. And we need to enrich these hardware accelerator with better accuracy and sensitivity. Of course, we don't want to discuss the high hardware cost or long development lifecycle for FPGA platform or GPUs, because when we are talking about life-threatening application or the need for very fast genome analysis, then the cost and the time for development might not be the most critical issues. All right, this is the last point. And as you remember that we don't have such machine to do the complete, to provide the complete sequence of genome analysis or complete sequence of a genome, but what if we have such a machine? Remember that read mapping is not going to die, or that this field is not going to die. All the efforts we spend in hardware acceleration can be used for other areas such as whole genome alignment, metagenomic, pangenomics, where you compare the complete genome to a complete genome. So we still have an open question, how and where to enable fast, accurate, cheap, privacy-preserving, and exabyte scale analysis of genomic data. So we don't know where to place the sequencing machine, where is the best to do such processing, whether near memory or near storage and so on. So I invite all of you to read our paper for more information, more details about the hardware accelerators for genome analysis, and read our very recent paper that got accepted in Genome Biology about read mapping in 111 pages. For more, you can check our lectures and other talks, other papers. And thank you so much, and I'll be happy to take questions.